Now here we are going to talk about the selectivity of the membrane. Okay, selectivity is like when we are making a selection for anything. Okay, you go shop or choose and you select the type of shoes you want, how much you want to spend, color. Okay, and we select students to enter the program. Okay, by looking at many different things. Okay, there is a selection process. Cell membranes do the same. They can discriminate, okay, between what they want to enter and what they don't want, or what they want to get out or get rid of. Okay, they can, for example, select or block substances depending on their size, okay, electrical charge or chemical properties. Okay, sometimes because they have negative charges, they are going to be repelled by the cell membrane. Okay, if they are made of anything that has any electrical charge, okay, it won't enter easily. Now, there are substances that freely enter into the cells, okay, that the cell membranes don't stop them ever. Those are small and non-polar molecules like oxygen, carbon dioxide, and many lipid-soluble molecules. Okay, basically, everything that is lipidic in nature will pass through the cell membrane with no problem. For example, hormones that are made from cholesterol, testosterone, aldosterone, cortisol, these steroid hormones will freely enter the membrane. Okay, but there are substances that are not welcome freely into the cells. Those are the large substances or polar molecules or ions. For example, glucose, sodium, potassium, any other ion, chloride, anything that has a charge will not enter the cells. Okay, amino acids won't enter the cells for free. Okay, they need to use specific transporters or channels. Okay, if the cells have channels or transporters for that substance, they are going to enter into the cells. Okay, there we have more examples. Okay, here on top, we have small molecules like oxygen, benzene, nitrogen. Okay, some substances that don't have charge, like urea, glycerol, carbon dioxide, ammonia, they freely diffuse. Now, large molecules like glucose or these ions, cations or anions, amino acids, nucleotides, they can't pass unless there is a transporter. Okay, on the right side, we have examples of it. Here we have a channel protein or a pole. Notice that this channel protein is an integral protein that is always open, okay, that has a pore in the center, that will have specificity for certain ions. We may have some of these channels that only let sodium enter. Okay, some other channels will let only potassium. Some channels only calcium. Okay, if you remember from your previous studies in chemistry, okay, there is something that we call the atomic radius. Then we have the ionic radius. So all the ions will have a different radius or diameter. They have a different size. Okay, so the channels are going to only allow these substances, okay, to enter through. Okay, we're going to be seeing more about these channels, how can they can be activated or inactivated, okay, how they work. And it's going to be, when we start talking about the action potentials, about the resting membrane potential, about how that is generated, maintained, we're going to be understanding more about these channels.